Hello everyone, I'm Shih-Chien from Taiwan and my nickname is Cherry. I'm 15 years old and a 10th grader. It's my second year studying at EF Academy in New York. I'm a girl who loves science, especially biology. It's a big honor to be able to stand here and present what I know. Today, I'm going to talk about a current amazing discovery for treating Alzheimer's disease. It's a type of dementia that was originally described by a German pathologist and psychiatrist called Alois Alzheimer in 1906. Alzheimer's accounts for about 60 to 70% cases in all types of dementia. And the common symptoms are loss of motivation, memory loss, and not managing self-care. As the disease evolves, patients' bodily functions are gradually lost and ultimately leads to death. There are many factors that could lead to Alzheimer's disease, such as lifestyle, environmental, and genetic factors. The development of the disease is associated with amyloid plaques and neurofibrillary tangles in the brain, although the ultimate cause of brain cell death isn't known yet. But the collection of amyloid beta plaques on the outside of the brain cells is a prime suspect. There are no efficient treatments for Alzheimer's so far. Some medications may alleviate the symptoms, but they do not delay the progression of the disease. Alzheimer's is also a costly disease. According to Alzheimer's Association, this year, there are nearly 44 million people worldwide suffering from it. And the global cost is estimated to be $605 billion, which is extremely expensive. In my presentation, I would like to introduce you about a new promising approach, which can potentially prevent the progression of the disease. In August 2015, an international team of scientists led by Professor Annie Kava from MIT had published a research paper which suggests a treatment for Alzheimer's disease. The investigation demonstrates a method for breaking down amyloid beta aggregate. Amyloid beta is a type of protein that forms insoluble plaques in the brains of Alzheimer's patients. So single molecules of this protein are not dangerous, but as they start to aggregate, they become toxic for cells and eventually promote cell death. Amyloid beta large aggregates are considered being involved in disease development, but soluble oligomers are also toxic. In order to achieve their ultimate goal, scientists have to break down the A-beta aggregates first, and they find a way to do it with heat. Heat is released when magnetic field acts on magnetic nanoparticles because of hysteresis effect. Heat is released very near of magnetic nanoparticles. That means A-beta aggregates have to be very near to magnetic nanoparticles. But how? The important task is to situate magnetic nanoparticles immediately to A-beta aggregates. Otherwise, they will spread throughout the brain. And if stimulated with alternating magnetic field, they will bring heat all over the brain, which is not desirable. Why not? Because it may cause serious fever. The nanoparticles, therefore, are coated with polyethylene glycol to stabilize them, and the special peptide sequence is attached to them to get the nanoparticles attracted to A-beta aggregates. The nanoparticles are so tiny, with only 20 nanometers in diameter, so that they can work efficiently without disturbing other tissues or organs in the brain. They are also made of iron 2,3 oxide. So with their composition and size, scientists can achieve efficient hysteretic power dissipation at physiologically safe alternating magnetic field conditions. There was research from other scientists saying that soluble oligomers could be even more dangerous for cells than large aggregates. However, our scientists realized that upon the treatment of A-beta aggregates with alternating magnetic field in presence of magnetic nanoparticles, they didn't actually get oligomers, but smaller pieces. However, they still want to know if breaking down A-beta aggregates could be dangerous for cells. So real hippocampal neurons were tested in vitro. So A-beta aggregates and the neurons were placed in one petri dish with the application of alternating magnetic field. And now let's take a look at the result. So here's the table for 4 groups that they are supplied with 3 hours alternating magnetic field treatment. The first one is the control group, which the cell viability remains almost 100%. And when they are added with A-beta aggregates, which the second column is shown, the cell viability decreased dramatically down to 70%. And the third and the fourth groups, they are supplied with magnetic nanoparticles and the polyethylene glycol. We can obviously see that their viability is increased like a lot. They actually didn't decrease that much. But I think the most outstanding result is the fourth column, which is the last group, that it is supplied with magnetic nanoparticles, the polyethylene glycol, and the peptide sequence. And its cell viability actually barely decreased at all. So I think which is absolutely amazing. Although the treatment for Alzheimer's disease still needs time to be tested and to ensure that it's secure enough to be as carrying out in humans. However, its concept has totally convinced me that its feasibility is potential, which may encourage more discoveries to enable protein disaggregation in physiological environments in the future. If so, by the time it's a chance for Alzheimer's patients to break away from the every mental impairment. Thank you.